Hello everybody. As you can see, I have an accent coming from Spain. I'm working on that, but sorry, you don't understand anything, let me know, I repeat the sentence. Okay, so moving forward, here. So these are, the title of the presentation today is HLA and Transplantation. It's an overview of the HLA role on the transplantation here in the United States. This is the disclosure statement. I have nothing to disclose, and also the planning committee disclosure. So HLA stands for Human Leukocyte Antigens. HLA is the human version of the major histocompatibility complex. It's expressed in all vertebrates, and they are glycoproteins. There are mainly two classes, HLA Class I and HLA Class II, and the genes are on chromosome 6. This is the structure of the HLA Class I. As you can see, there is, I think that I have a mouse here. There is an alpha change and a beta change. All the HLAs are a dimer, and the beta change for the HLA Class I is the beta-2 microglobulin, which is not polymorphic, it's totally monomorphic and is on chromosome 15. The HLA Class I is expressed in almost all your nucleotide cells, and under inflammatory conditions, it's only expressed in nucleated cells and in platelets. And the HLA Class I present peptides to the CD8 T cells. The HLA Class I classes are A, B, and C. The HLA Class II is also a dimer, alpha and beta, and the HLA Class II it has a much more restricted presentation or expression. It's only expressed in antigen-presenting cells, and is under inflammation. It's also expressed in other cells. Endothelial cell is one of them. And this is important for transplantation since they are the target of the antibodies. There is a dimer, it's a dimer alpha and beta change, both polymorphics, both on chromosome 6. And HLA class II present peptides to the CD4 T cells. And there are 3 different types, HLA-DR, HLA-DQ, and HLA-DP. For HLA Class I and HLA Class II, this area here where the peptide sits is the functional part of the protein. The function of the HLA is, as I'm saying, presenting peptides. Usually those are self-peptides coming from your self-proteins, and the T cell will recognize the HLA with the peptide and recognize that as a self-protein, as a cell that is healthy, and then will move to the next cells without being activated. But when you have an infection, like here I'm depicting a virus peptide, some of the virus proteins are going to be degraded, and those virus peptides are going to be loaded on the HLA and then presented to the T cells. So, and this is how your immune system recognizes infections, and this is how your immune system will be activated and will fight this infection. In transplantation, since mainly for solid organ transplantations, we don't really do a good job matching the patient and the donor HLA type. Usually the T cell will recognize the foreign HLA with the foreign peptide and will activate the immune system, and that will cause rejection. And that's why in every transplant you need to do immunosuppression. You have to give immunosuppression drugs to the patients, otherwise they will reject the organ. So the HLA is the most polymorphic system that we have in our body. These proteins are very, very polymorphic. The polymorphisms mainly are located in the pocket where the peptide sits. For HLA Class I, the polymorphisms are going to be in the alpha chain, domain 1 and domain 2. The beta-2 microglobulin is holding the alpha chain together. The Class II, the polymorphisms are going to be also in the same area where the pocket, where the peptide sits, and both beta chains and alpha chains, both are polymorphic. The exception to the rule, because there is always one exception, DR alpha is monomorphic. So usually we don't even type DR alpha, we only type DR beta. But the important thing here to remember is that HLA is very polymorphic. And this is important because this is the top of the protein where the peptide sits. So this is kind of like the view that the TCR will see. TCR will recognize this upper area with the peptides. These protein peptides are holding there in place inside the pocket by the forces that attract between amino acids. So if you change the amino acid composition of the HLA, since every HLA will have different composition of the amino acids, they will bind different array of peptides. And this is very important because what you want is diversity. You want to present to your T cells as many peptides as possible. This is HLA Class I presentation. This is HLA Class II. You can see that HLA Class II, the peptides is bigger. The HLA Class I is a little smaller, but the important is you want to present as many peptides, different peptides as possible. And this is, of course, we are, although hopefully living the pandemic time, but still, this is a paper on using, to transmit the idea of how different HLAs bind different peptides. This is an in silico assay where they took HLA proteins, they took the virus of the COVID-19, and make very small peptides and load it on the HLA. And then they counted how many peptides every single HLA was able to bind. You can see that A2O2 bind a lot of different peptides coming from the virus of the SARS-CoV-2 versus A25 barely bind anything. Like B46, that is a HLA-B that is very frequent in Asian population, especially in China, barely bind anything. It cannot present any peptide coming from the COVID virus versus B15 or 3, it presents several. So every HLA will present different array of these peptides, and this is the COVID, but you can do these experiments with any other virus and any other gem, and even cancer. There are a lot of cancer studies looking at neopresentation, and different HLAs will present different neopeptides. So everybody is more susceptible to a kind of infection or another kind of cancer. This is basically your immune system, and before medicine, your interventional medicine, this is what determines if you are going to be resistant to a virus or germ infection or not. So another characteristic of the HLA is the HLA, you inherit, it's in chromosome 6, you inherit one chromosome from your mother, one chromosome from your father. All the HLA sites inherited together are the haplotypes, and they follow Mendelian inheritance, where you can share one haplotype with any of your brothers in like 50% of the chances. No haplotype share is 20% of the chances, or two haplotype sharing is 20% too. So it's classical Mendelian inheritance. Also, another characteristic of the HLA is ethnicity or race dependent, in the sense that every ethnicity has a frequency of HLA that is different. For Caucasians, A1, BA, DR17 is the number one, is the ranked number one. So it's the one that you find more frequently, but it's much less frequent in Asians. It's ranked at the number 36. The most common in Asians, that is A33, 58, and DR17, it's much less common in the other ethnicities. So, and this is more important when you have a patient that needs a bone marrow transplant, since you're usually in these cases you are looking for perfect HLA matching. If you have an HLA yourself that is less frequent, since the NDP is biased through Caucasians, although there is a big effort to try to incorporate minorities and collaboration with like other registries from other countries, trying to mitigate this lacking of matching. So, but this is important for bone marrow transplantation more. But HLA, the reason why HLA is different between the ethnicities, there are different reasons, but it's because HLA evolved in different areas in the country. Now we have international fly-ins, and things are much more mixed up, but especially in areas more isolated, they tend to have the same HLA. So this is how diverse is HLA. This is up to mid this year, 2022. The number of alleles described for HLA class I are 24,000 and more. That translate to about 13,000 different proteins. For HLA class II is 9,000 alleles, that translate about almost 6,000 different proteins. And this is how it's almost exponential. At the beginning, the HLA was described with serology assays similar to the ABO, and these are less sensitive assays, so the number of HLA discovered was much smaller. But then, as the technology improved, introducing molecular testing, SSP, SSO, and then sequencing in 2010, things start to to be much higher, the rate of discovery. And you can see how many different we have right now. And now we have next generation sequencing and nanopore sequencing that allow for sequencing this whole loci area. So yeah, it's very diverse and it's not stopping. This is the number of new alleles that we have incorporated since last year. 2,000 class 1 new alleles, that is about 700 proteins, and 1,000 new alleles for class 2, that is about 500 proteins. So we keep discovering or describing new HLA because we have better tools. So in solid organ transplantation, the main thing, our main focus is antibodies against HLA. This is what causes rejection. And the question is like, how do you get sensitized against an HLA that is not your HLA? The natural way to get sensitized is pregnancy for the woman. We have about, the risk for getting to produce HLA antibodies is about 30% per pregnancy, so multiple women tend to have HLA antibodies. And of course, if you get a transfusion, you have a chance to get sensitized. Platelets express HLA class I. And although we do leukoreduced blood, sometimes there are some passenger leukocytes that can produce reactions. And transplantation, of course, we all know that if you don't, a lot of patients reject. They produce antibodies, and that is the main cause of rejection. These patients then need a second transplant, and then we have to deal with this high level of antibodies. It's been described also that as natural response against viruses and bacteria, sometimes there are some antibodies are produced that cross-react with HLA2. This is the interaction of an HLA protein with an antibody. As you can see, they are roughly the same size. This is HLA-A2, and you're looking at the protein for the side, by the side. So this is the beta-2 microglobulin, the alpha in pink, and in brown you can barely see that is the peptide. The yellow area is the area where the antibody interacts with the protein. This is called the structural epitope. At the center is the functional epitope, is the 1 to 3 amino acid that really is the target of the antibody, that is the specificity of the antibody. Okay? If you change the functional epitope composition, the antibody will not bind, but the structural epitope is the one that holds the antibody, stabilizes the interaction. So the same HLA, now you are looking at from the top, where you see the peptide here, the alpha change, and again, the area that another different HLA and antibody against HLA could bind. So this is the structural epitope in yellow, and then the functional epitope here in the center. Those are two different HLA antibodies binding the same HLA protein in a different epitope. And this is a very important concept. This is the concept of shared epitope. And before I move forward, EPLET is a theoretical epitope. It's something that if I'm looking at two different HLA proteins, there are areas they are different, potentially could be the target of an antibody. This is named EPLET. In solid organ transplantation, there are a lot of publications looking at a lot of different EPLETs between different HLAs. But the concept that I want to transmit here is the concept of shared epitope. To simplify the structure of the HLA, I put this cartoon where imagine you have a patient, and I'm only depicting here 3 different epitopes. And this patient gets in contact with this donor, either for it's a transfusion or maybe a solid organ transplant. And you can see that there is one eplet that is the same, and then there are other areas that are different. This patient can create an antibody against the different epitopes that they are not shared. So imagine that they create an antibody against this 24S. This antibody now will bind to the donor cells, but if this serum of the patient encountered a different HLA because you need a second transplant, it will bind to a different HLA, but the same epitope, that has the same epitope, and it will bind to this one, although the patient had never seen this HLA, but it has seen the epitope. If it creates an antibody against this other epitope, this is much broader expressed, more HLAs share that, so it's going to be binding to all these HLAs. So if this is a patient that lost the organ and we are looking for another transplant, at the end, the only compatible HLA is this one. It doesn't share anything with the patient. It's a very different HLA epitope-wise, but the patient doesn't have antibodies against this HLA, so it's compatible. So the idea is like HLA, basically, you have to think about HLA as a different epitopes combined in a different way, every single time is a different HLA. So we at the HLA laboratory, what do? We do HLA typing, and we do HLA typing for solid organ recipients, solid organ donors, for platelet recipients, platelet donors, for trial investigations, and sometimes for disease, drugs and disease association. We do the testing for molecular testing. Basically, we do real time. We are looking into next generation sequencing or nanopore sequencing down the road. That's something that we want to bring to the lab. But right now, we do a real-time PCR. And also, we do antibodies testing, and this is the main bulk of what we do. Luminex Technology, and I will explain a little bit what Luminex Technology is. It's very flexible technology, and we use it for detect antibodies against HLA, to identify the HLA that the antibody targets, and calculate the PRA. And then we do cross-matches. That is the final test that you do before transplant, and directly measure the compatibility between the patient and the donor. This is the Luminex technology, and I don't know if you're familiar with this technology, but it's very versatile, and there are different kits available, and it's based on the ability of having these beads, that every bead has a different combination of two colors, fluorescent colors. So each bead has a different combination, so it's unique. When this bead goes through the Luminex instrument, that is a fancy flow, it gets activating and it's bright in the specific bead, which the software captures and tells you which bead it is. For us, since we are interested in studying antibodies against HLA, this bead is recovered in HLA proteins. The main testing that we use is called single antigens, HLA single antigens, where every single bead has one HLA, one specific HLA. Since every bead has a unique color, the software is being built to identify this color means this HLA. These proteins are made in vitro through transfection and purification, and they are purified and loaded into this bead. This is what the manufacturer does, and it sells the kit. And then we took this kit, that imagine this is a bead with an HLA, we incubate that with the patient's serum, and if the patient has antibodies against HLA, we'll bind to the specific bead with the HLA that the antibody is targeting. And then we have a secondary antibody so we can see the reaction. And then this is how we see the reaction. This is after the 3-hour testing, this is the result. And you can see there is a line for the beads that had HLA-As, a line for the HLA-Bs, and then a line for HLA-Cs. This line here in the middle is an epitope. It's the BW4, BW6 epitope. It's the only epitope that is named by the software, by itself, because it's very frequent. It was described in the 60s, and you can divide the HLA-Bs in those that have the BW4 or the BW6, so you can characterize them. So what we do with this information, when we have these results, the thing that we need to do is to understand the pattern. So what usually we do is trying to see what is the actual epitope that we are seeing. And I'm seeing that it is an antibody that reacts with the 142 TKH epitope, which is expressed on A2, 68, and 69. There is another antibody that reacts with an ATI epitope that is expressed on 24, 23, 32, and 25. So I can identify. So for this patient, I have identified for sure that the patient has 4 different antibodies, potentially a 5th one, because I know there is a very common and very immunogenic epitope here. So, but this is important for us because we want to account for every single bead reaction, so we know we are dealing with a real antibody. Sometimes there is background reactivity, and that helps to understand, oh, this is background, I cannot account for an epitope, but this is real because there is an epitope. Besides that, we have like reference lines or reference cutoffs. We have these lines here that are telling us like anything that is lower than 2500 MA5, that MA5 is mean intensity fluorescent, then it's too weak, it's very weak, so we don't call it. And anything that is over that is really something that you have to avoid, so it's a strong antibody, let's avoid donors with these specific HLAs. We had different lines depending if we are talking about kidney, or we are talking about transfusion, or we are talking about thoracic heart. Hearts and lungs, this is the thoracic line, because these are sicker patients. They really need to get transplanted sooner. So it's much worse for them to be waiting for the perfect donor than transplant them, although there is antibodies against the donor type. We can deal with the rejection after the fact. So this is for a class 1, and then the class 2 analysis is similar. We have to do class 1 and class 2 by separate because the kits are separate. And you have a line for DR, a line for DQ, and remember here the DQs and the class 2, you have an alpha and a beta that is polymorphic, so you have to look for both of them. So it's DQ alpha, DP beta, DQ alpha, DQ beta, DP alpha, DP beta, and the DR alpha is monomorphic. We don't look for that, but in exchange, we have the DR 51, 52, 53, that is an extra DR with a different beta chain. And this is how it looks like when we have incubated the beads carrying these class 2 antigens with the serum of the patient. This patient reacted with these beads. Again, I'm trying to look into the epitopes that are really there, and this is the 3 epitopes I'm seeing. And I'm satisfied because I can account for all the beads. So that's the 3 antibodies that are causing this reaction. So this patient has antibodies against DQ alpha 3, 4, 5, DQ7, DQ8, and DQ9. So has antibodies that target the alpha change and the beta change of the protein. So those are 2 different antibodies. With this information, what do we do? With this information now we calculate the PRA. So you can go, one moment, here, again. And this is also the reference lines that we use. The same values for class 1, 2500 and lower at weeks, over 2500 and higher, and for thoracic we have a higher value. So then we calculate the PRA. You can Google OPTN calculator, and you can go to this page, website, and you calculate the PRA. In this case, we plug the antigens that we have recognized, because since a lot of times you are not sure about which epitope you are dealing because you have too many of them, as happening for class 1, you basically put the antigen that is target of the antibodies. And this patient is highly sensitized with a PRA of 96%. The PRA is the proportion of donors that carry at least one of the HLA that I'm putting here as not acceptable, unacceptable. And the PRA accounts for the frequency of the antigens. Like if you have an antibody against A2 only, the PRA is 47%, because 47% of your donors most probably carry the A2. It's very frequent. A lot of the people has A2, but A68 is less frequent, your PRA will be at 10%. Because this patient has so many antibodies or target different HLAs, the PRA is much higher, it's 96%. And basically it's telling you that only is compatible with 4% of the donors. A patient with high PRA has less compatible donors, and they will wait longer for a transplant. And if it's this, if we can do the PRA only with class 1 for platelet transfusion, and we at the blood center, we have a really big list of donors that are HLA types, so usually we can find something that is compatible. So as you can see, LSA is the assay that we mainly do in the laboratory. We do about 1,000 runs per month. That is a lot, because they have to be analyzed by hand, usually, and the directors are the only ones allowed to do this analysis. So myself and the co-director, we are the ones doing that during the week. So we do a lot of them. And it's good because you get familiar with the pattern. It's very interesting. So we use that in the pre-transplant setting to calculate the PRA, to identify the HLAs, and also you can monitor treatment. If a patient goes through the sensitization, you can monitor the level of antibody with this assay. Then at transplant, you can use that as a virtual cross-match that I will talk a little bit later what it is, but virtual cross-match, basically, you look at the antibody profile, look at the HLA of the donor, and you see if they are compatible or not. In the post-transplant setting, you do the assay, but you are only looking if the patient has antibodies against the HLA of the donor. That is called DSA, donor-specific antibodies. You don't care about the PRA at that time. You only want to know if the patient is rejecting. And also you can monitor rejection treatment. After pheresis, you can monitor if the antibodies level are dropping or they are not. You can use also LSAs to support transfusion for refractoriness in trials. And here and there we do some studies for neutropenias and thrombocytopenias too. The other tests that we do in the laboratory are cross-matches. We do cross-matches by flow. Serology has been the gold standard for many years, since it was starting in the late 60s. But in the United States, most of the laboratories are doing flow cross-matches. They all need serum of the patient. They all involve cells of the donor. And this is the tricky part sometimes, because for solid organ transplant, we receive offers from outside the state. And if we don't have the cells in the lab, we cannot do this testing. And then you have to fly the organ or the blood from the donor, get to Seattle to the lab, and start the testing. So it delays a lot sometimes with the offers that are out of the state. For serology, you will use complement. We all know the antibodies can activate the classical pathway of the complement and kill, and then you will see how the cells die through the complement cascade. And for flow, you use markers, basically antibodies against some receptors, so you can separate the cells. We use lymphocytes because the lymphocytes express HLA. We use these cells as a target of the HLA antibodies. These cells express HLA class I. B cells are antigen-presenting cells, so they will express HLA class I and HLA class II. And lymphocytes, as you all know, are easy to obtain. You don't have to do a biopsy. It's very easy. Ideally, you should be using the organ cells, but that's much more difficult. So since the lymphocytes already express the target of the antibody, we use lymphocytes. And this is how you will look into the results. For serology, you will need a microscope, and then you have a live dye that is telling you the cells is dead or alive. And for the flow, you can separate T cells and B cells with the markers, and then you have a negative control that is your background, and then you compare the histogram from the negative to the patient and see how stronger it is by comparison. So the difference is like the serology cross-match is much less sensitive and specific than the flow. And it was used, when it was in place, it was more like if the cross-match is positive, it's a contradiction for transplantation. If it's negative, you can go ahead. Right now, the flow cross-match is giving you much more granularity, so you have a number, and you can decide if the number is too strong or too weak, or you have a better idea how much reactivity is there. So right now, a positive cross-match is not a contraindication for transplant. It's only a consideration of the risk. The CDC cross-match was implemented in the late 60s, beginning of the 70s, as the gold standard, and it was to avoid hyperacute rejection, and only was implemented for kidney. And it was, since then, we have to do these cross-matches pre-transplant for a kidney. But currently, we don't have hyperacute rejection anymore. It's very rare, and if what happens, it's not due to HLA, because we already know by LSA, the Luminex assay, if the patient has antibodies or not. So the technology has changed, the cross-match has changed, even the treatment has changed a lot. So right now, what we have is a range of risk, low risk versus high risk. And we consider the LSA results, we consider the cross-match results, but also which organ we are talking about. Is a heart, is a kidney, and inside these specific organs is a good donor, is a low quality donor. Those are factors very important. Is the patient very sick, can wait or cannot wait? So we have much better idea of what is the individualized risk per every patient. And then we decide to move forward or not based on all these factors. It's not anymore a yes, no based on the cross-match. So that's all the field is advocating for virtual cross-matching, because when it's a local donor, the HLA laboratory will have the cells, we can do a cross-match, but we are sharing so many donors across the states that we need to have a tool that we can compare the antibody profile to the HLA of the donor and decide. This is an example of a virtual cross-match where you have a very highly sensitized patient, And for simplicity, let's talk only about DR. The patient gets an offer, DR11, DR17. Everybody can see that DR11 has antibodies, is very strong, the crossmatch will be positive. If the offer is DR7, DR17, both are negative. You can move forward with the transplant because the crossmatch will be negative. But then you have situations where you have something like that, the DR4, you have some very strong, some very weak. Here is when you probably need a physical crossmatch. So you can pick and choose when you're doing this crossmatch. And in this case, it's going to be dependent of the allele of the DR4 or the level of reactivity in the crossmatch that you are going to decide if you want to do this transplant. So virtual crossmatch is something that a lot of the HLA laboratories start moving and start doing, and we are looking into implementing that for next year. So the good things about the virtual crossmatch is it will accelerate the process. Now you have an offer from Florida or from another state. You don't have to wait to get the cells here to do the crossmatch. You can look at the profile and do it. So it will expedite the process. Also, you have to keep in mind that a lot of the deceased donors that we are having are the elderly compared with previous years. The population is getting older, so the quality of the organs has decreased a little bit. So ischemia time will affect more this organ than an organ from a 20-year-old person. So speed is very important. The problem with virtual crossmatch, as I mentioned, you have an allele-specific antibody, an antibody that's very weak that you don't know how it will react. And also, I have shown you that there are so many different HLAs, like 20,000 different proteins. The HLA assay has 100, less than 100, about 90-something class 1 and 90-something class 2. So it not covers the whole thing. It covers more than 90% of the most common epitopes, but there are little holes. So sometimes you can deal with that. So patients that are highly sensitized, it's not recommended to do virtual crossmatch, but non-sensitized patients, you can move forward. The problem is CMS regulation, that they are still stuck in the 60s where we have to do that, although the tools have changed. And ASH is in conversation with CMS trying to change the policy. So how the allocation work in United States? So when there is a potential donor here in our area, we get the blood, we extract DNA, we do HLA typing, and also we do ABO typing at the blood center. This is how we do typing, real-time PCR. It's 90 minutes, it's super fast, and it's only low resolution, but it's very easy. And our techs in the middle of the night, they don't really need any input. They can do the interpretation by themselves. Then this donor type is introduced in UNIT. UNIT is the website that is the database for all the transplant in the United States, where all the patients are there also with all the information about the HLA typing, antibodies, and other things like the size, weight, what kind of organ they need, and characteristics that they accept on the organ. And then there is a match run where the donor type is crossed with all the database on recipients, and a group of recipients is selected. From this group of recipients, based on how long they have been waiting and the quality of the organ, different factors, and every organ has different allocation policies, so it's not for everybody going to be the same, but more or less there are different factors, that is the quality of the organ and the patient that they have to match. Then we will do either a virtual cross-match or a physical cross-match to finally assess the compatibility between the donor and the patient, and then the transplants happen. The selected patients will get the heart or the kidney or the liver or the intestine that they are waiting for. UNOS and OPTN are the ones that create the policies and manage the database. UNOS is a nonprofit that is under contract with the federal government. They manage the database. They are always on call. They are the ones that facilitate the transplants and the allocation and do the match run, and ensure that the policies are followed, and educate the public and the community and the transplant community. So they are in charge of that part. And OPTN it has committees of physicians that they are looking at what is need to change, create the policies, and then the board of directors approve those policies and implement those policies. And they work together, so we have good policies that the transplant center are following. This is how many people is waiting for an organ transplant in the United States. More than 100,000 people waiting. This is a chart from that ends in 2017. It's a little old, but you can see it was higher and it's dropping slowly. And this is because we are incrementing the numbers of donors and transplants. And this is what we need to keep doing. We need to increase donors and we need to increase transplant. The only way you increase transplant is you have more donors. This is 2021 data. We had about 20,000 different donors and we facilitated about 40,000 transplants. And I don't know you here, but we had made this year the 1 million transplants in the United States, so it was a big deal. So for kidney, you can see that this dark bar is the number of kidneys that we transplanted in 2021, about 25,000, little less, but 90,000 people was waiting for an organ. For liver, the disparity is lower, but still 12,000 people waiting, only 9,000 transplanted. For heart and lung, there is not that much disparity, but those are sicker patients. They either die waiting for an organ or they are dropped from the list because they are too sick to get transplanted. Every 8.5 minutes, we add another person to the waiting list. It's about 200 per day. 17 people die waiting for an organ every day. And from all the people that die in the United States, only 3 out of 1,000 die in a way that can be a donor, because you have to be in a hospital, in a controlled environment, and then you can be a donor. This is how UNOS and OPTN divide the country so it's easier to manage. They divide the country in regions. We are region 6. Every region has at least one OPO. OPO is the Organ Procurement Organization. It's the team that go to the hospitals where there is a potential disease donor and deal with the family and with take care of the donor and take care of the family and try and harvest the organs. Here in the region 6, we have 2 OPOs, one in Seattle, Life Center Northwest, and one in Portland, Oregon, that is called Cascade Life Alliance. They just changed the name. We have here in Seattle 5 transplant centers. University is the bigger one. We do here thoracics and abdominal. Children's, that they do livers, kidneys, and hearts. Swedish, they do livers and kidneys. Virginia Mason does kidneys. And VA has a very small lung program that basically works with UDAP doing the transplants. The blood center, Northwest Northwest, we do the testing for all these patients. Then there is a transplant center in Spokane that has another HLA laboratory, and then Portland has one laboratory taking care of 2 transplant centers, and Hawaii has 2 transplant centers too. Note that neither Montana, neither Idaho, neither Alaska had a transplant center. Alaska patients go to Seattle. This is so far at September 5th, the number of donors, and that is living donors and deceased donors. There is a big push for living donors. You can be a living donor of a kidney, and you can be a living donor of a liver, half of the liver. UDAP is doing those, starting I think like 2 years ago. So as a United States as a whole, as September 5th, we had about 14,000 different donors that facilitating 29,000 transplants, but we have added 47 people to the list. So we are adding more people than transplanted people. Locally in our region 6, we have had 500 donors, transplanted 886, but also adding more people to the list than people that we are transplanted. As you can see that donors can come from everywhere. Even we have one living donor from Illinois, but the transplant happened in Seattle or Oregon or Hawaii. The transplant center is where the transplant centers are. So we are in track to have the 20,000 donors that we have usually per year, and more than 40,000 transplants, but still we have a deficit because we have more people waiting for transplant than organs available. Here's an example of how the allocation works geographically, and every organ was a little different. I'm only showing kidney since it's the main organ that we transplant. We have a much bigger list. Since last year, we have moved from only sharing inside the area, inside of the zone, that we are, the area, to these 250 nautical miles. So if you have a donor, these organs usually go first to the 255 miles recipients that are in the area, based on different factors, but it's primary to this, and then if nobody accepted there, then it goes to other organs. Since we are in a corner in the country, we don't have that much competition. A donor here in Seattle will be shared with Spokane and Portland, but Portland will share only with Seattle and Spokane also with Seattle. Portland and Spokane are too far away, outside of the 250. But you can see in the East Coast how competitive this is. There are too many transplant centers too close together, so there is much fair competition for the organs. But if you don't have a recipient in that 250, you ship the organ to the other states. So we constantly are shipping organs to the different areas, so flying them in regular flights. So the important thing here is like the organs, when they are harvested, they have a limited time. They don't last for days. Hearts and kidneys, hearts and lungs usually about 6 hours or less, pancreas less than 18 hours, and kidney can last to 36, but that's a lot. So how we do solve this problem, since we have to fly them, and usually the flights, we know that going to Florida is not a 3-hour flight, so it's far away. So usually this is how the organs were transported. It's basically a cooler. It's ice and the organs were inside. This is how, for years, we have been shipping the organs. We are moving to these technologies. Our OPO use Transmedic, and they ship the organs. When they need to ship the organs far away, they use this system. As you can see, it's much more expensive. But now a kidney can last 3 days. It's not 24 hours. You can ship them far away. And also you can monitor the functionality of the organ, because you can collect the liquid that is going through the organ. It's a pump pumping some media inside the organ. So we have moved from here to there. It's still not for every kidney. If it's a local donor, usually it's more like this. But if you have to ship them far away, then they try to put them in pumps. And this is also better for the organs that are not as good because they are coming from elder people. Then this is much better for this kind of organs. So, and now to finish, only like a case that it had a good ending. That was a 27-year-old woman that had 4 kids. In the last pregnancy, she had a postpartum cardiomyopathy that needed a transplant. She couldn't survive without a new heart. The bad thing is that the patient was highly sensitized, 92% PRA, and this is only with these antibodies here, this. If you have to add these weeks that are not counted because those are not unacceptable, the PRA will be 100%. So she was 92 officially. So we had like a week to find a donor. We couldn't find a donor for her. She was too highly sensitized. So they did a total artificial heart for her. And then we had more time. During this time, it was like 3 months or 4 months, she got several offers. All the time they came up positive all the time. So we couldn't find a good donor until finally she got this offer. I circle here the 2 that are mismatches. You can see that everything else matches the patient type. You don't create antibodies against your own HLA. You create antibody against HLA that is different. So basically, she only has 2 HLAs that are different. And you look at the PRA, she doesn't have any antibody against the donor. You only have to look for A1 and A3, and it's not here listed, meaning that she didn't have these antibodies. So we did the crossmatch, and the crossmatch was negative, as we were expecting. The crossmatch was negative. So the patient got transplanted. The patient now is July 5th that we saw the patient, and here we monitor for the HLAs that are different. Since only are 2 different, we monitor for these 2. The patient is negative for antibodies against the donor. The patient is technically not negative. The patient has antibodies. This is her reaction. But boxed is the donor type, is negative. The donor type is negative, so the patient doesn't have DSA, but it has antibodies. If she happened to lose this organ down the road, for whatever reason, she will be sensitized, and we will go back to square one, trying to find an organ for her. But thankfully, she is sticking to the organ is fine. When you have such a good match, usually immunosuppression is good enough and you don't reject these organs. So that's a happy ending for her. We had to do a lot of cross matches until we found the perfect match. So that's where I end my presentation. So you have any question? Yes.
Hi. I have a question. The question is, how does HLA play its role in, say, for example, autoimmunity?
Yeah, I mean, HLA also because present peptides. And you know, like how thymocytes are matures in the thymus. And HLA is part of the training of the thymocytes that end up being the lymphocytes. So this is where you can delete the lymphocytes that carry the HLA, the TCR that recognize the auto-HLA. Those are eliminated. But sometimes this is not perfect. So sometimes you have leakage, and this is what causes autoimmune diseases.
I recently learned that Bloodworks offers a test called the Advanced PRA. Does that also test for HLA antigens? Or is
Fast PRA?
Advanced.
Advanced PRA. No, I mean, what we do as the PRA, have you seen that in the menu for the HLA laboratory?
I think so, yeah.
So we call PRA, and the PRA is the calculated PRA. And we have two ways of doing that. One is a screening assay that is telling us if there is antibodies or not. It's a qualitative assay, yes, no. And if it's no, that's it. And if it's yes, then we do the single antigen assay. And that's the two tests we do. We do another test named DSA that is basically the same assay, the interpretation is different. It's only interpreted as post-transplant if there is the antibodies against the donor. The assay is the same. So basically we have three modalities, and sometimes we have also the option to order that STAT, that means fast. It means that instead of having like a 24 hours turnaround or 2 days turnaround, we can do a 6 hours turnaround. We have techs on call. We have techs on call every day. So we are on call 24/7. So we can always call a tech to do the technique.
This is a question online. In patients with high PRA, can you either induce tolerance or suppress or block anti-HLA? And I ask for you to repeat the question as well.
Okay. So for patients with high PRA, if we can induce tolerance or suppress antibodies, we cannot induce tolerance. And I mean, there are some studies inducing tolerance for kidney transplants where you do a bone marrow transplant, and then trying to induce tolerance using the donor cells. That's kind of a high risk, and they have working very well, but it has a lot of risk. For patients with high PRA, they do desensitization. Basically, usually is pheresis, IVIG, and drugs like rituximab or bortezomib, other things trying to reduce the PRA. You can do that in pre-transplant, trying to reduce the PRA so you can get an offer, or it's the same treatment for the rejection. If you are rejecting, then you can use these drugs to try to reduce the reactivity against the donor. Yes?
So I saw in one of your earlier slides a reference to sort of NGS as an emerging technology, or sequencing as an emerging option, and particularly to the long-range rapid sequencing modality of nanoflora. I was wondering if you could talk a little bit about sort of what are obstacles to implementation of sequence-based.
Yes. So he's asking about the complexity of implementing next generation system in our laboratory. We are looking into that right now. The complexity is, I mean, it's expensive. For solid organ transplantation, there is not the need, because the policies right now are that you have to type the donors a low resolution, meaning A2. I don't have to know if it's an A201 or A203. So there is not the need per policy, but if you want to identify epitopes, you need to have the high res. The complexity is like the timing is more expensive. You have to have for us in particular, we don't have the space, we have to locate the space to put the instruments and training and everything. The next generation sequencing, it takes about 2 days to 3 days to get a typing. You can type a lot of people at the same time, but it takes 2 to 3 days. Real-time PCR, it takes 90 minutes. The alternative is a nanopore system that is coming to the market, which there is one only company offering that, and it's 6 hours, but also it's much more complex than doing a real-time PCR. So the analysis is more complex, and since in solid organ transplantation there is not a policy that makes you do it, a lot of laboratories that they don't do bone marrow, they are still not doing next generation. But we all know that that's going to be the future, that's something that to make sure that the donor and the recipient are compatible and be able to do virtual cross matches, accurate virtual cross matches, we need that technology.
Kat Borstein says, I'm curious if you could talk more about the transmetic systems. How do they work and what's the magnitude of the cost?
The transmetic system, so she's asking about this transmetic system to the pumps where you put the diseased organ and you keep that functioning longer time. I don't remember the cost. That's also is something that the OPO, the Life Center Northwest, has them, and I know they're expensive, very expensive. And also, you have to have a specific team from Transmedic doing the shipping, because it has to be very specialized. You have to control the viability of the organ, you have to monitor, so it has to be specific people trained for that do the shipping. So I don't really know how much they cost. I know they cost a lot.
Somebody else is asking, actually, a follow-up from that other person without even knowing the follow-up. Do you know what's monitored? Is the transmetic system, what do they monitor in the organ, and do you know anything about that?
I don't know exactly. Every organ is going to be different. So for hearts and lungs, we monitor one thing. For livers, we monitor enzymes. For kidneys, the urine output, these kind of things. Down the road, in the future, there are talks about you can treat certain things, but that's something that is coming. How you change the preserved material, that the liquid that you are injecting, you may be able to treat some problems that the organ has. So that's coming in the future. I don't think that we are right there. But you can monitor the certain values. Yeah. Yes?
Blood work class, HLA types, some certain platelet donors. What determines what donors blood work class HLA types?
We type them in bulk, in the sense that when our pool of donors, HLA type donors, is getting too low, then we start asking to the platelets donors that come in voluntary, they want to be typed. Because if they want to be typed, it means that here and there we call them, we need you have to come to the center, you have a window of time very short because we need you for a patient. So it's only when we have low, and I think the last time we did that is like 3 years ago, where we needed to type more patients, or maybe 5 years ago, we need to type more donors. So we have incremented the pool, and we were looking also for minorities, because we have a broader Caucasian population that is good for Caucasian, but sometimes minorities, they don't have, because you're looking for HLA match platelets, and as I explained also, the different ethnicity races have different HLAs, so sometimes we have holes, so we tried at that time to type minorities. Right now, my understanding is that we are good for a while.
And one more question from me, and that's if you are, I know there's been staffing issues, and when you're trying to keep a lab staffed 24/7.
Yes.
Pandemic, has it been challenging?
It's been very challenging. We just finished a round of we hired 4 people. We are very trained tech left last year, and we had to promote internally, and it was really, really hard to find techs. We have to do, motivation with increased salaries and things to bring people.
What are the requirements for the techs that are
We only require a bachelor in science, and we train them, because nobody trained in HLA, not even in the university, you don't get trained, not even the medical laboratory trains in HLA. So we hire them usually as a bachelor fresh from university, and we start from ground zero to train them. So it takes about 3 years to train a tech, so they can be independent, because when they are on call, they basically by themselves at the lab doing the technique, and there is another tech at home, a very senior tech, they can consult with. And then they consult results with the director. So the directors, we are always on call. Elena and I, we share weeks. 1 week she's on call, 1 week I'm on call. So we are always on call. Yeah. But yeah, it was challenging with retention and hiring. I think that we all had the same issue. Yeah, we hire 4 people, so we are hoping they are stick, and it's gonna be smooth. Okay, thank you very much. Thank you for hearing.